Cell-cell interactions in the extracellular matrix. Types of cell junctions. Cell structures include membrane organelles as well as non-membrane organelles, vesicles for transport, and structures that extend from the cell's surface. Membrane junctions are composed of both integral and peripheral membrane proteins that function to connect and support cells. The main function is to attach the base of a tissue to the extracellular matrix. So we have epithelial cells that are bound to the basal lamina for structural integrity. Some of the components include keratin proteins, integrin proteins, laminin, fibronectin, and of course collagen. This gives our cell structural rigidity as well as support. As you can see in the image, different kinds of structural components keep the cells in place and aligned. The red proteins are called dystonin, the green proteins are called plectin, and then attached to them are different variations of collagen. These will then bind to the laminin and attach to the base collagen fibers that give structural support to the tissues. The first type of junction is called desmosomes. These are also known as macula adherins. Macula adherins connect cells together through a thickened structure called a protein plaque that contains many filaments that extend between the two cells. They provide structural integrity to the cell that allows them to deal with any kind of mechanical stress. Some examples include the cadherin protein as well as the intermediate filaments. Cell number 1 is over here on the left side, cell number 2 is on the right, and you can see these little green proteins that glue and anchor them together. Adherin junctions. Adherin junctions' main function is to attach two adjacent cells together in a rigid fashion. This allows the intercellular space between the two to house fluid and act as a passageway for certain substances that can be transported between the cells. You can see on the left-hand side the E-cadherin domain bound to the right cell, which also has an E-cadherin domain. Some examples include actin filaments and cadherins. Tight junctions, also known as zonula occludens, prevent any kind of intercellular cell leakage by attaching each cell via proteins called occludens. They also prevent the mixing of the plasma membrane and lipid on either side of the cell junction to maintain the cell's polarity. So you can see that they're closely linked together, not much room for any passageway. Cell number 1 on the right-hand side and cell number 2 on the left-hand side. Gap junctions form that provide a direct passageway from one cell to the other cell. Some examples of the components include connections. These proteins include 6 parts that form a small tubular tunnel between two adjacent cells. This allows molecules to move between them. So the composition of the extracellular matrix is very complex. We're going to have cell membrane components. We have structure components, including the cytoskeleton. We also have cholesterol embedded inside the membrane that gives flexibility and fluidity. We have collagen, as well as different kinds of polysaccharides, fibronectin, and integrin proteins, as well as receptor proteins. Cell fate, the phases of the cell cycle. The cell cycle is the division of somatic cells resulting in the creation of two identical daughter cells. The two basic phases in the cell cycle include interphase as well as mitosis. A cell spends the majority of the time inside interphase, where it's focusing on maintaining itself, carrying out metabolic activities. And during interphase, it consists of several phases: G1, first gap stage, S stage, or also known as DNA replication, and then G2, second gap phase. During G1, the first gap phase, cells begin to grow, produce new organelles, and they form structures necessary for DNA replication. During S phase, the 46 double-stranded DNA molecules are replicated, forming two identical sister chromatids. During the second gap phase, G2, cell division enzymes are produced, centrioles are replicated, and it gets ready for cell division. Then comes the mitotic M phase. This is where we have the prophase, metaphase, anaphase, and telophase. During this time, the cell is going to go through division, splitting the chromosomes in half, resulting in two identical cells being formed.